see many of these seminars, and I find them very useful and warm, and I'm happy to be giving one. So let me go ahead and share screen to start. Let's see. So I can start a third. And switch over. Okay, I think people can see this now.
Yep.
Yeah. So I'm a grad student at the University of Michigan and have been working with the MSU Detroit Partnership for Food Learning and Innovation, or DPFLI, with the director named Edwards, who has also worked in the same research lab as me. And so we've started on this project about biochar, and so I want to talk about sort of some of the things that I've been reading up on along with our approach to it. So first, I want to share are the resources that I've used, just to be sort of open where the information is coming from. So one is a book called Terra Preta. It's relatively kind of it's a casual read that hits on many different facets about climate change, previous agriculture, and how you can even try and make some biochar and the potential for biochar. And there's also this other book called Amazonian Dark Earths, which I'll talk about, which are the original biochar soils. And these are sort of written by some of the researchers who popularized biochar studies. And of course, the U of M library is giving access to a lot of original research articles. So the outline for today, I'll be doing a sort of a simple structure, is introduction, and then talk about the origins of visions for biochar, and then talk about some ways that people have measured properties about biochar with methods, some results from other studies, and the studies we've been doing at the MSU DPFLI, and then we can open it up for a discussion. And you can also ask questions in the chat. So to start, I want to start.
Can I interrupt for 1 second? Nick, I'm sorry. Can I interrupt for 1 second?
Yes.
So I just want to, just to reiterate to everybody, if you have questions during the presentation, please, you can put them in the chat, and then I will help, navigate those with Nicholas as he's presenting. So when there is a natural break, we can, ask questions and so forth. Sorry about that, Nick. Go ahead.
Yes. Whenever you'd like to ask a question. And so, yeah, I want to start in the Amazon basin in South America. It's mostly in Brazil, but it also touches on some regions in the surrounding countries like Peru, Venezuela, and Colombia and Guyana. And so here it just talk about the discovery. This is a map of some of the areas where archaeologists and soil scientists have found some of these Amazonian dark earth sites. They're in squares. These are just some of them, but there are a lot more. There are also the names of some of the indigenous groups, which some of this will be a sort of an indigenous story, sort of along in part in honor of Indigenous Peoples Day today. So they were originally recorded, these soil profiles, in 1875. And then just as a naturalist with a Cornell professor and a student, then they were re-recorded a century later in 1980. And then less than 20 years later repopularized by these three scientists. And so what the finding was is that usually there are these soil profiles you can dig, and they are typically nutrient poor. And the reason is because we think of a lot of biodiversity and lots of life thriving in the tropics where it's wet and hot. But in these soils, there's much fewer nutrients because all of the nutrients are instead locked away in the bodies of organisms, but not actually being stored. In other words, there's a lot of decomposition that is quickly getting recycled and being used, which is what you want for plant productivity. And they're called Ferrocells because ferra comes from the Latin iron. And so they have lots of iron that is combined with oxygen. And so they have this reddish color usually. But instead you find there are these really dark profiles that have darker colors beyond the spectrum of what's normally found. And they go pretty deep. And what they're finding is usually just coincide location-wise with archaeological sites for archaeological research. So this was generally of interest. And the darker colors usually comes from organic matter, which comes from slower decomposition. And so instead of the nutrients being quickly accumulating in more life and more plants, it's being, you have less decomposition, it's being stored away in soil. And so that's an interesting distinctive feature of how tropical systems operate. So in all these sites that were found in different parts of the Amazon basin, mostly in the western side, there's some observations of fragments. There's micro charcoal. This is what we know of as burned material. There's the ash that happens when that burned material is completely allowed to decompose in the fire, and those are rich in nutrients. The charcoal provides a black color. There's pottery, which are fragments of pottery, and there's all different types of pottery, and so that's the archaeological side that was found. There's also some fish bones, which is indicative of habits that indigenous peoples were using for food and hunting, and remnants of human feces. So you find it in different ways down the profile in these piles. There were different practices, but part of the question is what's going on and how did they arise? And there's a lot of variation in where these profiles were found. So on the left, the right part, the brightest orange, is a typical ferrocell that has the red color from the iron. And those are nutrient poor, and you find them next to those. You find the dark earths there, and you also find them on the other side of the fertility spectrum nearby, these are nittocells. They have clusters or a stronger cluster formations or aggregates, and they're not yet as weathered and bright as the ferrocells. So they're much more fertile, and they're used for coffee, and they're considered pretty fertile. So there's variation in the starting soils. There's also variation in the activity, the inferred activity about it. So in the middle bottom, there's a picture of a dark earth that is more of a gray color, and that is colored more like terra mullata. And so the famous example is terra preta meaning black, and terra mullata is more of a mixture of black and white, so it looks more gray. And so there's variation in what the inferred use was, or the terra pretas were more of, they originated from combining human waste from villages, and these were used more for the result of fire after consecutive seasons of cultivation in the same place. So there's slightly different ways that that the profiles develop. You can also see on the bottom right, this is another typical profile where it just has so many shards of clay and the pottery. And so that serves another function. The one on the right is affected. You see fewer shards on the top, and that is because of current ongoing agricultural use. They're still being used today by modern indigenous groups. But these shards served to redirect water in the soil, change where the air was, and they sort of help promote the buildup of organic material and nutrients. And then on the top, you can see there's lots of variation.
Deeper than I am.
In the pottery, the types of pottery. So there were 4 major groups in terms of uses: cooking and serving, pots for eating, and then for transportation. We have similar, types of things that we use: serving bowls or eating bowls. But what's interesting is you can see that they varied by indigenous group in the region. And so that would be an interesting question that is sort of more difficult because it's about the past, but how do different groups, how did their different terra preta profiles vary based on their pottery and their practices? One thing that was noted in common, this is showing a diagram of a hill slope that affects soil development. It's called a cantina. Most of the dark earth profiles were 15 meters high about. And this is important because it rejects the possibility that these were formed just by natural landscape processes, where you would think that because wetlands are considered ecological supermarkets, they're super productive, that people would settle in the much closer to the river, but there's a flood risk. And so they're actually way up high, and that is more indication that these were anthropogenic and human caused rather than naturally occurring. And so this is just a graph. I bring this up because this is from this year, that evidence about the hypothesis that it's not actually human caused, but the humans arrived after there was already fertile soils. So what this is showing is that it's dating different carbon. In the bulk soil, that's the straight line, that's the solid line. At different parts of the soil, different depths of the soil profile, and then the dotted line is the carbon from fire, or pyrogenic, and they did this for adjacent soils and for the ADEs. And what they see is that they dated the carbon from fire way long ago, before the oldest ADE was occurring.
What's ADE?
These are the Amazonian dark earths.
Okay.
It's the name of all the terra preta, all the profiles with dark horizons. And so this was leading them to say, because I put some dates in here for reference, humans arrived in the Americas 11,000 years ago. Then ceramics, the oldest were 7,000 years ago, and the oldest dark earth profile was 3,000 years ago. And so this may suggest that the biochar is actually really much older than when people were actively cultivating, using it for agriculture. So it might not be human induced, but this was responded to later in the year, more recently, that the focus should be focusing on the topsoil and how humans are changing that, and its natural and much less likely that humans will affect deeper down in the soil. So most of human effects are just in the topsoils. And so it's still, the idea is that they're anthropogenic. And even more evidence is the spatial organization of where these profiles are found. This is an example village center that has a circular formation. Sometimes you would find the dark earth profiles in the center, depending on where people gathered and where people put their refuse. But these are in the backs of houses. And so these dots are showing that there are different amounts of organic carbon. And so the highest one, that's 13.5, is back near where the dark earth profiles are. So this is pretty clear evidence that they're human made. And these super fertile soils are, this was a really interesting indigenous technology. So the inference about the use, I put this here to sort of show the pores and the solid parts of soil. And the pores are important for releasing some greenhouse gases and other gases getting as a byproduct of metabolism. But the biochar was used to block these pores and essentially limit decomposition that way, because a lot of the microbes that do decomposition, they're fungi and they breathe like us, oxygen, and then release CO2. And if they don't have oxygen, then they can't actually break down what's available. And so this use of ADE technology was also thought to allow higher populations over 10,000 and grow rather than using shifting cultivation. You're allowed to use semi-permanent. So this is sort of like, you can think about it as a composting system. Some people refer to it this way, where you have your greens and your browns. The greens are rich in protein and nitrogen, and they're wet and they're fresh, and they're fruit, they're rich in sugars too, so they're readily decomposable. And the browns are more slower to decompose. So you have the dead leaves and the cardboard and the paper. The biochar in this case was more like the browns, where it's harder to decompose. And so it's essentially preventing and regulating the decomposition of the greens in the soil. What's interesting to note is that they also had fish bones in these dark earth profiles. So that's one thing that we are thought to not include in our compost. It's not recommended, but that's an interesting sort of anomaly there, and that they seem to have done it, and it's in part responsible for the high calcium content there, which is definitely human made. So biochar in a way acts to regulate decomposition when you have it very high and affect how decomposition happens in soil. This is an example of slash and burn. So this would be in contrast to the semi-permanent. It depends on how you use this. So first you have a fire and you clear it so that you can cultivate in a season. And you can either do short rotations, which are 2 and 5 year, or longer rotations, which are over 10. And the longer the rotation, then the more time you allow the soil to redevelop. This data is showing increase in aggregation, or the amount of clods in soil, which are important for physically limiting decomposition. And so the more of that you have, the reservoir for nutrients and biodiversity. So over time, biochar allowed for the shift to permanent and cultivation or longer fallow periods. And finally, as this technology was being developed by indigenous peoples, there was the end to its development, where you had invasion by Portuguese colonists, that their perspective was coming in and really just extracting what is possible and using human systems of interaction that were disrespectful and just violent and promoting slavery. And so it's unfortunate for the development of this technology. And most of the end was caused by disease, which were differences in immunity, in part because of the proximity of large mammals. But there were many different groups that were doing this alongside. There were contemporaries, and it stopped around 500 years ago, the 1500s. So after this exploration, there were a lot more profiles found in the Amazon basin. You can see in the inlet, but they're also found in many different places. So there's dark earth type soils in central Mexico with the Aztecs. There's some in South Africa and Liberia. And there's also Nordic dark earths, like I showed you before, that were more gray that were found here. So this is clearly something the dark earths, though the method of development may have been different, it sort of converged around the world, which is interesting. So from this, now you know sort of the history, you can continue to ask, how do we understand this better? So how is it exactly that dark earths were made, and what combinations of feces or biochar do you need? How much does biochar, the dark earths, affect agriculture? They're a lot more productive. And can we use ADE technology to combat climate change? So I want to talk about some methods that we're using nowadays to get at these. So originally, well, first the original production, you needed to produce biochar with fire, but with low oxygen. So this is piling wood and enclosing it with either soil or some kind of other enclosed mix of branches and soil, so that it could burn. You have some airways so that you can allow some airflow, but not too much to keep the fire going. And that's how it was originally made. Sometimes you can pile it up or dig into the ground and put wood there and then cover it. Yes, but essentially you want it to burn, but in an enclosed way. Currently, there's a gasification process where it's more industrial. And these containers, instead of being made of soil, they're of metal, and you put the starting wood and you burn it, and you have a channel so that you allow some gas to escape. And the gas that's produced is rich in its hydrogen and some greenhouse gases. And that gas can be connected then to another digester that can make biofuels that way. And then at the bottom, the byproduct of this gas production is the biochar. It's the burned wood that was burned in low oxygen. So most of the biochar comes from staying as carbon. If the fire were to happen in an open area, it would turn to ash. So biochar is in a way the stage before ash that is rich structurally, but in fewer nutrients. And these are some suppliers. This is the one that we use, Wakefield Biochar. You can get some cheap small amounts or large ones, depending on what you would like to try. And some measurements that are relevant for biochar. This is the ribbon, just showing the ribbon technique for soil texture, which should be affected by biochar. A lot of the site at MSU, the DP-Fly, is part of it is clay and part of it is more sandy. And so this affects how much biochar you want to use. So it's relevant to understand what the texture is of your soils. One of the things you can measure is the porosity. So one effect of this burning with low oxygen to make the biochar is that the wood is only partially decomposed. So you retain a lot of the open structures and the pores. And these are showing, this is with a scanning electron microscope. This is on the order of 50 microns. So it's medium bacteria and single hyphae strands of fungi. But each of these pores, I think that is important for the ecology of microbes in the biochar habitat. Each of these can support a population of bacteria, and it also affects how fungi grow through the soil. These are also places where you can store water and store nutrients. So I think these pores are a really important function of how biochar improves soil fertility. There's also moisture. So in those pores, biochar can retain moisture, and the moisture generally in soil functions in that it's a different, you reach different moisture levels when you're in the wetting stage versus the drying stage. So if you have a really dry soil, you might start up here. I don't know if you can see my mouse by the top left corner, where it's very tightly bound to the surfaces. This could be biochar or just a soil particle. And a plant root is not going to be able to get at that because it can only exert so much force. It gets the help of fungi and mycorrhizae, but even they have a limit. And on the other extreme, there's the pore-filled region. So when you have a lot of water, then these films are sort of wider and the water is more easily accessible by the roots and the fungi. But this focus on drying and wetting patterns is of interest for how biochar affects soil functioning more broadly. And there are a variety of tools you can use to measure in-field moisture or in the lab. Some are, this is on the more expensive side, but there are also cheaper tools too. Another one is a pH and acidity. So biochar is known for usually increasing pH or lowering the acidity. And this affects potential nutrient availability. So how you have an acidic soil is that generally in any soil, a clay particle usually will have has negative charged surfaces, and on those bind the nutrients. So calcium and potassium, which are used in cells for different functions, and those are usually bigger than hydrogen. Hydrogen is the smallest atom, and so there's sort of an exchange that happens where just by normal metabolism, roots, they breathe like we do because they don't have light available, and so they opt for the respiration pathway instead of photosynthesis. So they will either produce a hydrogen that will be in the soil water or CO2 that will react with water and then end up producing this hydrogen, and those can exchange with the clay to then free up the nutrients. And so the more hydrogen that you have, then the more acidic your soil. You also may have heard of activation for your biochar. This is because biochar is mostly carbon and not other nutrients. And so the biochar needs to be combined with either a liquid source of nutrients or a solid source like compost. And compost is really rich in phosphorus, which is akin to the ADE soils. So that would be a good one, and that's what we have done at the site. So you can just combine this in a bucket with either compost tea or worm castings or urine or some manure, and you mix it, and here it says 12 to 24 hours. I've also seen 2 weeks, but usually the longer that you do it for, then the more ready it will be. There's also, so we've touched on the moisture component, the nutrient components, then there's the microbial activity component. One way of measuring this, the most common way that lab tests do it, either at Kellogg or Cornell, they will incubate some soil and have one jar with regular soil and one jar that has been exposed to chloroform that kills all of the microbes because it's toxic, including to us. And so then they will measure how much CO2 is produced during regular respiration and then compare those. After adding a little bit of inoculant, like something that's easily digestible, like sugar. There's also this new microbiometer that I sort of heard about earlier in the year that is newer and being used for classrooms. It's more simple than the lab incubation method. It's you basically just take a soil and mix it with water. You add a little bit of something easily digestible like sugar, and you wait 20 minutes. And then you pipette out some water and you put it at the center of this color wheel. And based on the darkness of that liquid, then that's you have relatively higher or lower amounts of microbial biomass, which is usually very good for making nutrients available and so on. So this is all part of getting to understand how it is that biochar works and some of the variables and components that would be important for explaining how biochar works. So in addition to just microbes in general, there's fungi specifically, or mycorrhizae. These are the fungi that attach to roots. One way of surveying them is to look at their diversity and DNA. And so this, on the upper left, I have an example of a simple DNA extraction that can be done at home with strawberries. I did this one time. It's the same principles as what's done in lab. You can take a strawberry and mix it up, and you add 2 tablespoons of this extraction solution as just detergent. So it can be dish soap, and you keep mixing it, and this breaks up the cells. And then you filter out all of the cell debris. These are proteins and the membrane. And then in the remaining liquid, you drop some alcohol, and that alcohol goes around the DNA, and it winds up into something that's visible. So this same idea is done also for PCR tests, like for COVID tests. So comparison to the antigen test at home, if you send some saliva into a lab, then they'll do the same thing. They extract the DNA in the same way, and then this process of amplifying the DNA strands. So it's just heating and cooling repeatedly, where you heat the solution so that you separate the DNA. You prime some areas where the enzymes combined, and then you heat it up so that enzymes can do their work. And that's just this 3-step process over and over again. It's basically DNA replication, which we do all our time in our bodies to grow, heal tissue. So that's one way of looking at, it allows you more specificity for the DNA to look at different species of fungi, or if you want, bacteria. And then there's the next level up, which is looking at soil invertebrates. So with respect to biochar, there's little work done on this in general because soil tests tend to be more physical and very chemical. But in terms of the ecology side, these larger organisms are of interest. So you can look at these by sieving manually to a sieve. They will you will easily see them. They will start crawling, and you can aspirate them into a little vial, suck it up. There's also using this light gradient, a heat gradient. So these bulbs on top, if you put the soil into a regular funnel and then just some mesh so that the soil sits, then over time, over a couple days, then the invertebrates will walk out and go into the bottom of the tube where you might have some alcohol and preserve them. So this is called a Burley's or a Fulgrim funnel. And generally there's, once you have that data and those organisms, then you can start to analyze and think about, well, which are the groups that are doing most of the decomposition work? Which are the ones that are eating fungi that are, mycorrhizae on the roots, or they're ones that are actually eating the roots, and those are pests, or they're ones that are just predators that are eating other taxa that are pests themselves as a form of control. So there's certainly more to explore here. And so now I can, before talking about some results after methods, to say what we did at the DP Fly is this: we set up an experiment with different particle sizes of biochar. This is going back to the surface area and pore availability of the biochar itself. That is important for the ecology of microbes and invertebrates. And so we had none, these are the controls, just adding our mix. This is regular biochar as it comes. Large sizes, which are over 1 millimeter, and then small, which are less than 1 millimeter. And so when we applied it after tilling 4 inches, this is what it looked like. You can sort of see the gradients. And so I talked generally about some results. So moisture, here we found some significant effects of particle size on moisture retention. So this is, in part, this study allows to explain how is it that the biochar is working. Well, it's actually important to pay attention to different sizes play different roles in the soil. So the mixed, the normally applied biochar had a few percentage points higher soil moisture than the small particle sizes, and the large one was the same as control. So it's best to apply the biochar as is, but the explanation for why you get more retention is because of the smaller sizes. And so that makes sense with higher surface area and more places where the water can bind.
Did you have a control in this study too?
Yes, this none was the control.
Okay. Can you explain what those mean? Those 4 letters mean?
Yes. The N is none, so it's the control, no biochar. The M is a mixed size, so it's really just not unaltered biochar. that was just applied to the plot as it came. So mixed sizes. The S is small sizes less than 1 millimeter, and then large sizes over 1 millimeter.
So large size biochar did not, it did not help the soil moisture content.
No, yes. Most of the function of moisture came from this small particle size.
Okay.
So, yes. This nutrient availability, this is going back to original studies from ADEs. One of the notable facets of ADEs and one of the arguments for why it is anthropogenic is that there's much higher phosphorus and calcium. There's also higher nitrogen, but that nitrogen is not as available for plants to use. The nitrogen is usually what we put in for fertilizers that we get, and usually you really see quick increases, but in the tropics, phosphorus is more of a limiting nutrient. So this is just,
I'm sorry, excuse
Yes.
me. Would you say that, generally speaking, would you call biochar like a sponge of sorts that helps the soil with holding on to nutrients and water?
yeah, I think that's a good way of saying it, because the surface area is really important. So that's sort of what sponges provide. They hold a lot of water, more than, clays are small and they have lots of surface area, but their shape is different in that it's really flat. And so when they combine together, they sort of form these dense packs, and water can only go through parts of it, through like the bigger pores. And so they form, they may form when it's dry, sort of cracked films at the surface, like you may have seen, and it's very hard. And then when it's wet, it's super, mobile. Whereas biochar is kind of more balanced in its extremes, in how it deals with extremes. It will retain water when it's really dry, and not retain as much when it's really wet. So that's sort of, I think that's one goal. And when you have soils that are in urban settings that have not been fallow, if they're recently experienced change for development, like the site was, it used to be a school, then those soils are optimized for structural stability, and they will be more extreme in their temper. And biochar can, along with organic matter, can sort of mitigate that a little bit. Sort of my broad picture. So yes, this is just to show that it has higher total nitrogen, ADE, and phosphorus, but only the phosphorus is more available. And that has to do with how the nutrients bind to the clay surfaces in the exchange with hydrogens in the previous image I showed. Microbial biomass, there's also more in ADEs compared to adjacent soils. This is from a previous study, and this is showing an image just so you can see how fungi, these are their hyphae, and the glues that they exude. Actinomycete is it's a actinobacteria. Oh, this is a hyphal. It's a hyphal microbe. This may be a bacteria. But they sort of enmesh themselves in that whole pore, and so that allows more space for microbes to grow. And this is a graph showing colony forming units. So this is using just growth cultures of microbes on a plate. And there are more numbers of colonies in ADE soils. compared to adjacent ones. This is also one way that biochar works. So it's a sponge for water and for microbial biomass too. There's also microbial diversity that was also been higher in ADEs. When you get to the diversity, there's a little bit less, there are a little bit fewer studies. And studying biochar in current context will be different than the ADEs themselves. But one group to note is this Acidobacterium, which lives well in acidic conditions. There is more diversity on the right in the ADE side, where you see two different lineages of groups that are evolutionary distinct in this tree, whereas just one related group in the adjacent soil. And this is going back to the another level higher is the invertebrates. And this was one study that was done at our field site with a master's student that I worked with, Seneca Lee. And so she took different samples with pitfall traps. So this is putting plastic containers flush with the soil level, just digging out an area where they can stay, and you cover them with a plate so that they don't get flooded when it rains. And you leave them for up to a week in the field. After that, you collect and identify all the different groups you have by order here. And you can divide them into different functional groups. So mesofauna, they're still big. They're less than 2 millimeters. And macrofauna, which are bigger than 2 millimeters. These are going to be the predators. And we found some difference in the number of species. And these are the same letters. Sorry, slightly different order, but there's a difference between the N, which is the control, and the M, which is mixed. So again, applying the biochar as is has a stronger effect, but in this case, it's a stronger negative effect. And so there was what helped explain this effect is seeming like both large and small particles equally. And so this is, it's not the most positive story that we would assume, because biochar has so many benefits. But there's much less work. The top left graph is just showing that the green line, biochar and fauna, there's much fewer studies that are published about this. So how soil invertebrates might be affected by biochar is variable, still unclear. Certainly how they're affected and the processes that make it happen. A separate group of studies that might be relevant for this is there's some toxicology studies in freshwater systems where pollution of degraded oil components these are carcinogens. They can also be produced by dissolved biochar components, or when biochar gets dissolved. And so that toxicity could help explain this loss of species. But there's much more ecology too. That kind of toxic effects would probably be more general to all the biology. But in terms of loss of species, that there's a lot more interactions at play there. So it's worth continuing to look into.
It looks like the variables here is I mean, they look pretty similar, like they're all close to 5, right? They're on the low end of 5 to 7 or something.
Yeah, so the effect is not large. It's is what we, but is statistically significant with what we found. So it might be that this, you might not see this effect at an adjacent site that has been fallow for a long time, and you have you know you have many weeds and organic matter, whereas this site was less vegetated because it was is being freshly revived after the loss of the school. So I think the soil context, the vegetation context can also affect what you might see. But I think I will encourage people at different farms with different histories to try this and see if if they get similar or different results, because in terms to be able to generalize that more studies are needed. This is, again, all in pursuit of a potential tool for combating climate change and doing climate smart agriculture, sustainable agriculture, but also in helping explain how is it that the original ADEs worked. A lot of their nutrient availability comes from the movement of particles and nutrients by invertebrates. That's what's thought, part of what's thought for their fertility. So this would be relevant there. And toward the end, what everyone's interested in is crop yield. So these are some original studies. The dark circles are Amazonian dark earths. The white circles are they call it transitional soils. They're the terra mulatas that I talked about before. So they're the grayish ones compared to the much darker ones. And so corn seemed to grow much higher pretty quickly compared to the adjacent soils, the Ferrocells. And in pots, again, corn, this was also, the adjacent soils also did worse compared to both the grayer soils, terra mulatas, and the terra pretas. So this is evidence for corn, but there's so much more diversity to grow. Yucca, that's not up here, but anything that you might want to grow, leafy things. Because moisture is such a strong effective biochar, like your brassicas might do pretty well also, I would hypothesize. And so there are a lot more species to test out and try adjacent to corn or okra. I think there are a lot more ideas to be tested that people can try on their farms. And at the end, we can sort of go to a discussion and questions. I just want to summarize some notable points is that biochar for agriculture and cultivation was an indigenous technology in the Amazon and is trying to be revived today with our more technological or industrial infrastructure. But we should keep in mind that vision from ago. And that the dark soils, the strongest evidence for their human, the anthropogenic cause is the phosphorus and calcium. And those dictate that they were possibly an intentional form of land management and composting. It's possible that it was unintentional. It is hard to know, but we just piecing the evidence together. And for the future, I would think that want to keep on asking what are the strategies and practices we can do to mimic what indigenous groups were doing centuries ago. How does biochar affect the ecology of soil invertebrates and organisms? So again, the previous studies were very chemical mostly. And so we can add more of the biology and ecology in there. What are the best contexts to use biochar? So there are lots of different crops and polyculture systems that could be tested out. That we need more space and more sort of, more instances of research to do. And how can we make biochar the most sustainable, given how it's produced nowadays with the gasification process that is industrial and mechanical. But we want to sort of cycle the whole, make sure to use the gas, make sure to use the biochar from the wood, and make that whole process have a sustainable life cycle. So thanks everyone for coming. We appreciate you coming and look forward to talking.
Great. So I definitely want to open it up for questions. There is definitely, I think people are really curious about, well, people are curious about production methods. Tracy, if you want to, if you want to come off mic, or off of mute, you can ask your questions if you'd like, or if you want to put it in the chat, either way.
Okay, thanks. I had only had it muted because my dog is barking at everything that moves outside. Okay, so I had a quick question about the shape of the dome that they created the biochar in. So does shape matter? Because there was one diagram that showed it like a dome, and then there was another one where it was sort of angled, and the wood was piled in at an angle. So does that matter insofar as creating biochar? And can we do that on our community garden land? Can you burn like that without getting in trouble? So those are my questions.
Yeah, so I don't, from what I understand, I don't think that the angle matters, just the covering matters more, but you do need some kind of opening. So it can be in the middle if you pile it up. But probably the angle in that diagram helped, for airflow, because hot air will rise.
Absolutely. Okay. And something we can do on our land without like having the fire department come and go, what the heck are you doing?
Yeah, so I know of one person in the Detroit Urban Farmers Facebook group who says he makes it. Once I found out that there are ordinances against compost piles that are too high, for example, I would imagine that there's some kind of policy also for fire. And so that is, I think that's an important point that I would like to dig into what, the city ideally should allow us to do that, but I think it takes more. It would just take more convincing.
Nick, do you have a sense of like how big have you done any research on like practical, like DIY, do it yourself kind of ways of making it?
Right.
So there is a group at U of M that made a gas fire, which is basically like a metal like garbage bin that was closed. So they sort of welded some metal pieces as a lid, and made an opening at the bottom where they could access the biochar. But basically, you would have you start with the frame of a metal trash can and close it and have an escape tube, and then a feeding area where you can put in a light and also the wood, and then at the bottom you would get more of the biochar. And the idea is to close it enough so that you get less ash and more of the biochar before it turns to ash.
So almost like a rocket stove, right? Do you know what a rocket stove is that you make with the cinder blocks?
Not specifically that one, but yeah, it's analogous to what people call carbon in Latin America. Is used for cooking too.
Yeah, absolutely. Okay, yeah, okay, you guys, I am so sorry. I gotta run. Thank you so much. This was really interesting, and I do want the recording, and I want a copy of the chat.
Okay,
Thank you so much for coming. Your
All
question.
right. Thank you. You guys have a great day. Stay safe.
bye, Tracy. So so would this be just, I just do give any kind of clarification, because people, this sounds like charcoal, so do you know how to differentiate between, just using standard charcoal, or what's the difference, and why? My understanding is
Yeah.
that you can't use charcoal, but I don't know, if you could help us understand why.
So in principle, it's similar. That the main difference is use, and so when you buy it, then it will affect how it's produced. So biochar sometimes comes like pre activated, where you have nutrients sort of interlinked in the pores already, instead of combining with compost. And the charcoal, like what you might want to use to start a fire, will come like lined with flammable substances that are more toxic, probably toxic to us and life and microbes that wouldn't help grow. So what we were using is part of why we use Wakefield is because it's sourced from pine wood. That's softwood, so it's probably more porous, but it didn't come pre-activated or anything. So in principle, it's the same, but like at the production and buying side, then you might get things that have already been processed and are, will take away from your use if you buy charcoal versus biochar.
There's a question, any particular wood, using any particular type of wood better than another for making it?
Yeah, there's a lot of interest in the wood source, the density of the wood, and the burning temperature for researchers. Again, that's less on the ecology and productivity side, but well, actually, it's a little bit ecological in that later, like denser species like maples, they are going to be less porous, and they have a different ratio of like oxygens and certain organizations of oxygens at the outer edge of their particle, compared to softer pine wood. So there is work on that. It probably depends on what you want to grow. For the softwood, if it's more porous, then I would suspect things that are more water heavy, that would be better to grow. Your lettuces and leafies.
What was the name of the brand of stuff that you used?
Wakefield Biochar. That one, there's also another one, Terracool, that we're looking into. I think that one comes pre-activated. They have activated options and a compost blend. They sell, yeah.
Can you spell that?
TERRACOOL. Yeah, and so they will have different options, either a compost blend or pre-activated or not activated.
And what kind of what rates were you applying it at? What's the recommended rate to apply it? Like how big were those
it depends
squares?
on your soil texture.
Okay.
The squares were they were 5 feet by 5 feet, and we applied at 5% by volume. So this would be however much compost you wanted to apply, then 5% of that.
Okay, So practically speaking, how much compost did you put down?
We put, it's, I think around half a bucket on a 5x5.
Half a bucket, and then how much biochar in that bucket? Like just like a half cup?
Actually, I think it was, I have to look back, but it was a half and half compost and biochar.
Oh,
But
okay.
that
So-
total amount was 5% of the soil block represented by the plot. If
Okay.
you were to dig,
5 feet down.
dig 20 centimeters down,
20 centimeters.
so like a foot.
Okay. So,
So it's not more than like 1 or 2 buckets per plot. And what's been used at it for other research studies, which is part of what we wanted to also be in line with, seems relatively small. It depends on if the study was done on sandier sites. They might need less, and if they pre-sieve it to a really small size, it might get more functionality for small amounts. But now we were testing much more to like 20% by volume. So originally the ADEs, they have to have, I didn't mention that they even have their own classification system. That's how much variation there was. And they need to have at least 10% by volume of the charcoal particles. And so but that was built up over hundreds of years. So it might be that the annual application rates were relatively small. But of course, we don't have hundreds of years for climate change. So we want to amp that up.
yeah. Can you elaborate on how, or if you'd said it, maybe I missed it, but if you could just reiterate or elaborate on how the production or how the use of these biochars is good for helping stop time, or could be good for help stop climate change.
Well, the idea is that instead of is that you can put more use to the dead leaves and wood that otherwise would just take a long time to decompose. And so when it decomposes, you get much more oxygen produced than is the remaining solid carbon that you end up with. But when you produce biochar, you have it stay more as carbon and produce less oxygen.
Okay.
So that's more of it.
And so is there any discussion? Is there anybody around the world that's doing this on a large scale for that purpose?
Yes, I didn't mention there are initiatives, the International Biochar Initiative, that pulls together networks of people trying to produce and use biochar sustainably. They have a website. It's just internationalbiocharinitiative.org. And so they're working on those kinds of making sure you use the gas for energy production, along with saving biomass for biochar production and making that system of production more sustainable. Also using different feeds, like in coffee agriculture, you might want to use the coffee husks. It depends on what you grow, how much, what the material is that you have available.
There's a question in the chat from John. Hi, John. What about cost, say per bucket of biochar? Is it fair to say that most of the fine size biochar that is commercially available, like what you use, would be cost prohibitive for small scale farmers?
They have 1 pound and 5 pound bags that would be like $5 or $10.
How much is a 5 pound bag?
You mean how big?
No, you said, so wait, is it 1 pound is $5 or $10, or 5 pounds is $5 or $10?
I was just looking at, I think maybe 1 pound is like $4.
Okay.
And then 5 pounds is more like 8. So now in terms of adding 5 pounds, I think that could give you maybe like 2 beds if I were to quickly estimate
Uh-huh.
2 raised beds. So I'm not sure what the current cost of input is, or how that
Yeah,
affordable
but
that
if
is.
you were to do that on scale and you try, so it could add up quick. Does anybody
Yes.
sell it by the yard?
Yeah, so then when they sell by the cubic yard, then it gets to be like $800
Yeah,
dollars.
wow.
So this is more, and there's there's one study that I know of that just used like a handful of biochar that tries to more efficiently use it right near the roots. Also, Naeem has done this with a separate study on the site, just to use it more efficiently, and get more bang for your buck.
Yeah,
But it's a matter of whether you're interested in more direct short-term crop productivity, or if it's a longer paced soil remediation thing.
right. Like kind of in combination with other things that you're doing.
Yeah.
Judy is asking, could you use straw? Could you use straw to make biochar?
Yeah, you can use straw to make biochar. I don't know the final properties. They will differ a little bit compared to wood-based, but yes, there are studies on those. Also applying straw and doing like lasagna mulching and biochar, that can also be used in combination. The straw would serve as the brown, and the biochar would also serve as a brown that holds more water.
Okay. Well, I think we'll open it up to, I think that's most of the questions that I had. Does anybody else want to share any questions with Nicholas? Hopefully we get to a point where this is, something that municipalities start to think about in terms of, what if all of our curbside tree trimming pickups and leaf pickups all went to a gasifier, and then we had biochar available. Wouldn't that be amazing? And but
Yeah.
um,
And the ideal is very local production rather than even regional, clearings. You could sustainably manage forests that are further away and then send and ship the biochar, which is what we did. But the most ideal is, yes, exactly what you're saying.
yeah. So last call, any questions? Okay. Well, I think we can wrap things up from there then. Thank you so much for your time, Nicholas, and thanks for sharing all of your research and your presentation. We appreciate you. We will be I'm recording today, so we'll put this up on our YouTube channel in the next few days here. And Nicholas, do you have any resources that you might like, other further reading materials or articles that you might want to share with the group? If you do, if you wouldn't mind emailing them to me, and I can share with folks later.
Yes,
I have a really stupid question.
Okay, ask it. It's probably not stupid though.
Okay, I have a 24-inch deep, 4-foot wide, 8-foot long raised bed. Can I dig a ditch down the middle of it, fill it full of straw and sticks, cover it with compost and burn it and make my own biochar? Would it be helpful at all?
So I think maybe you could, based on like kind of these old school ways, like, and Nick, please join, please chime in, but I would say we didn't dive deep on production. I think it would be prudent to get more detail on how to do it on a home scale. and maybe have some more practical design on how to do it before I would say go ahead with that. But I think you're on the right track. If you dug a hole, like that seems to be the way to do it. So Nick, do you have anything to add to that or correct from what I said?
No, I agree. And that if it's not too, if there's, no rule against it, then you can try. Why not?
Yeah, I mean, you can have in theory, you can have a bonfire, so it seems like along the same lines, though it may be, yeah, I don't know enough. I mean, maybe,
I just don't want to wreck my garden.
Mm.
yeah, I mean, I don't think I would do it in the same place as your beds, if honestly. But again, maybe there's, I'll take a look. Maybe there's some practical books out there that I will share also with the follow-up on the video.
That'd be great. Thank you.
Also, the book I mentioned, the beginning, Terra Preta, talks some about how you can make it at home. That's like the third section about it too. And that's a popular book that you can get at the library.
Say the name one more time, please.
Thank you.
It is called Terra Preta. It's there's a subtitle. Where is it? I have it. Let's see. How the world's most fertile soil can help reverse climate change and reduce world hunger, with instructions on how to make the soil at home. So it's kind of long. I can type it in.
I think Terra Preta will probably get them there.
Yeah.
Put that in the chat. So then there was a request about slides. Would you be willing to share a PDF of your slides?
Yeah.
Okay.
I can send those to you.
Okay. All right. Well, with that, I think we'll wrap things up for the night. Thanks everybody for coming out. And we'll see you at the next one. Thanks again, Nick. Appreciate you.
Thanks so much for having me. I appreciate it.